let's go ahead and get started with the DNA, RNA notes that we're doing. All right. So what do you need to know on this page? Well, okay. So you looked up your 5 facts about DNA and found out that DNA is made up of nucleotides, which is the monomer. We learned that back in our biochemistry unit, and that it's a double helix, and that there's a base pair ruling, and only certain nitrogen bases go with others. So that's kind of what we're going to focus on during these notes today. All right, so first thing, what is DNA? Well, it's 2 single strands that are put together a lot like if you could untwist this, you would see it looks a lot like a ladder. Okay, it's got 2 side rails, and then it's got the foot rungs in between. So if you can imagine that. In fact, the model in the classroom, I've got the first part of it kind of standing upright so that you can see that, how it's two side rails and then the rungs. That's what we call the double helix. And the nitrogen bases are what come together to make up the rungs of the ladder, and they're held together by what we call a hydrogen bond. So now you've got your ionic bond and your covalent bonds that you learned back in physical science. We talked about the peptide bond when you build a protein between amino acids, and now you have the hydrogen bond, which is what holds the nitrogen bases together as the ladder rungs for the DNA. There is a specific rule called the base pair rule that says adenine always pairs and bonds via that hydrogen bond to a thymine molecule, and guanine always pairs and bonds to a cytosine. Okay. There is a small little change in there when we talk about RNA, which is the other molecule we're going to look at. This thymine will go away and something else will be substituted in its place. So if you remember the little baby word at, you will always know the base pair rule, because that's exactly what it is. Okay. The molecule, the nucleotide, is the monomer, like I said before. Okay, and what does that look like? Well, it's a phosphate group, it's a sugar group, and then it's a nitrogen base. Here's your nitrogen bases for DNA. For RNA, remember I said there was going to be a little bit of a change? RNA does not use thymine. RNA uses uracil. It's one of the ways that we can tell whether we're looking at a DNA strand or whether we're looking at an RNA strand. If you look at the strand of letters and you see a T in there, you know it has to be a DNA molecule. If you look at a strand of letters in a sequence and you see a U in there, you know you have to be looking at a RNA strand. So what does that mean? Well, DNA stands for deoxyribose and RNA stands for ribose. So that's where the DNA and the RNA come up with. So the full name of DNA is deoxyribonucleic acid. RNA is ribonucleic acid. The sugar group, we've seen this before. The phosphate group, we've seen it before. And ironically, we've seen these same elements in a different configuration in our ATP molecule. Okay, so different situation, but same base elements. The nucleotides are broken into 2 subgroups. It is either known as a purine or a pyrimidine. Okay, and it's based, how they're broken up is based on how many rings the nucleotide has. So if it's got 2 rings in its nitrogen base, then it's a purine. And adenine and guanine are both considered purines because they both have 2 rings. If it only has 1 ring in its nitrogen base, then it is considered a pyrimidine. And cytosine and thymine are considered pyrimidines. Okay, so that's just some FYI, good facts to know. All right, how do you put all these pieces together that we've looked at? You know, we looked at the parts of the cell, okay, and that's where it starts off. And we know that the genetic material, the chromosomes, are inside the nucleus. Okay, all that is familiar. We went through all that with our cell structure and function unit. Well, if we take those chromosomes and we look at them in greater detail, you all probably have come to suspect chromosomes are in that X configuration because you've seen them that way, you know, all through all your education so far. But what actually happens is, if you can take this X configuration and you can uncoil it and uncondense it, you would see the spiral coming apart, just like you see in this visual here. And what happens, you'd find out that the DNA, the genetic material, is actually coiled and condensed around a protein called a histone, which is one of our vocabulary words. So it's a protein, and it works to keep the DNA organized so that it doesn't knot up, basically. So if you've ever taken an extension cord and wrapped it around your arm so that you can, or your Christmas tree light strand so that they don't get all twisted and messed up, that's the same procedure that you're seeing here. The histone is like your arm, and the DNA wraps around the histone to keep it from knotting up or getting all massively coiled. Keeps it neatly organized. And if you keep stretching that coil all the way out, you would get to what I told you, that ladder configuration where you've got the side rail and then the foot rungs going between. Okay? A segment of the DNA that actually codes for your characteristics, like how tall you are, what color your eyes are, what color your hair is, what color your skin is, those segments are called genes. So the DNA is made up of many genes on many of the different chromosomes. RNA, just slightly different. Okay, and like I said, the way you know that you're looking at an RNA strand is if you see a U in there for uracil as opposed to the T for thymine. Okay, but otherwise everything else is the same. It forms a hydrogen bond between them. It's made up of the same nucleotides. That all looks very similar. This was part of our homework today on Friday, so we'll look at it more on Monday, but the central dogma is what we term the three processes that occur with the DNA and the RNA, or the RNA. Replication is all just about DNA. It is when the DNA makes an exact copy of itself. And that's when I was coming around in the classroom and I was showing you my model and how it splits apart and then it makes an exact copy. That is replication, and we're going to do that in detail on Monday. If that molecule splits apart and those nucleotides come in and the uracil is used as a nucleotide instead of the thymine, well, you're looking at making what we call the messenger RNA strand, and you're looking at the transcription process. And those two things are both going to take place in the nucleus. Replication when the cell needs to divide, and transcription when the cell is going to make a protein. Okay, so it's going to make a protein. The how part we'll look at when we make our model. Remember I told you we're going to have movable parts so that you can see how it actually moves. The next step in the protein synthesis after transcription is translation. Okay, and translation is where the code is actually transferred and the protein is made in what we call protein synthesis. And this process takes place in the cytoplasm on a ribosome, which is an organelle we learned about back in our structure unit. Okay. One of the mistakes that students make is they think that replication leads to transcription and transcription leads to translation. That's not it. Okay. The central dogma is just highlighting the three processes that could occur to the DNA throughout its lifetime, basically. So replication is just when the DNA needs to make an exact copy of itself so it can do cell division, which is our next unit. Transcription and translation are the two steps that are going to lead to protein synthesis. Okay, and that's where RNA gets involved. Okay, so that is where we are at. This is a picture that shows you how the DNA replicates, which is what we're going to do on Monday. Okay, so let me walk you through the steps a little bit more detail than what I did on the table with each group individually. So here you have your parent strand, and you made a parent strand. That parent strand is step 1 is going to be unzipped, and it's unzipped by an enzyme. You see the ASE. ASE typically denotes an enzyme. Okay. Helicase is going to be the enzyme that's going to unzip the strand. Okay. As it then unzips, another enzyme called DNA polymerase is going to come in, and it's going to read the code. So when it sees an A, it's going to bring in a T. When it sees a G, it's going to bring in a C. When it sees a C, it's going to bring in a G, and so forth and so on until it builds the complementary strand. So when you're finished, you have two identical DNA strands. This one is probably going to stay in the original cell, okay, as part of the original cell, and this one is going to go off and in cell division become part of the new cell. What we end up with then is two cells that have exactly the same DNA in them, which is what we want. Otherwise, you're going to end up with a bunch of funky cells in your body, and that won't be good, okay? Or it may not be good. So that is the replication process, and that's what we're going to look at on Monday. And then after that, we'll look at transcription and translation. So enjoy the rest of your weekend, and I will see you in class on Monday. Bye.